Compounds that contain the sulfhydryl group, or the SH, are called thiols. They are similar to alcohols in structure, but the sulfur atom replaces the oxygen atom. Thiols have significantly lower melting points and boiling points than alcohols because the sulfhydryl group is much less polar than the hydroxyl group. Earlier, we learned that the hydroxyl group is very polar because the electronegativity of the oxygen atom, which is 3.5, is significantly higher than that of the hydrogen atom, which is 2.1. The electronegativity of the sulfur atom is 2.5, and thus the electronegativity difference between sulfur and hydrogen is rather small. As a result, the sulfhydryl group is only slightly polar and thus only exhibits weak hydrogen bonding. This is reflected in the lower melting and boiling points and lower solubility in water of the thiols in comparison with alcohols of similar molar mass. Thiols and many other sulfur compounds have nauseating aromas. They are found in substances as different as the defense spray of the North American skunk, onions, and garlic. The structures of the two most common compounds in the defense spray of the striped skunk, trans-2-butene 1-thiol and 3-methyl-1-butane thiol, are shown here. These structures are contrasted with the structures of the two molecules that make up the far more pleasant scent of roses, geraniol, an unsaturated alcohol, and 2-phenylethanol, an aromatic alcohol. The IUPAC rules for naming thiols are similar to those for naming alcohols, except that the full name of the alkane is retained. The suffix thiol follows the name of the parent compound. So for example, when we're looking at the IUPAC names of these four thiols, if we have ethane thiol, you can see that we take the ethane group, this is our alkyl group, and we add thiol on the end. Now, we don't have to put a locant or a number for the thiol. We don't have to call it ethane 1-thiol because it wouldn't matter which carbon it was attached to, it would still be the first carbon. In the second example, we still have our ethane, but now we have two thiol groups, and we have to indicate which carbons they're on. We have one on carbon 1 and one on carbon 2, and therefore we call it 1,2-ethane dithiol. Here we have another example. Here's our longest hydrocarbon chain. I'll circle it, so that would be a butane and then a butane thiol. The thiol is on carbon number 1, so we call it a 1-butane thiol, and then we have a methyl substituent at carbon 3, and therefore the name is 3-methyl-1-butane thiol. And we have another dithiol over here. Again, we find the longest carbon chain, which would be a pentane, but it's a dithiol. And we have a thiol group at carbons 1 and 4. And so it becomes 1,4-pentane dithiol. The amino acid cysteine is a thiol that plays an important role in the structure and shape of many proteins. Two cysteine molecules can undergo oxidation to form cysteine. The new bond formed is called a disulfide. And so here's a new functional group for you. The disulfide is alkyl, then we have two sulfurs followed by another alkyl group, the disulfide bond. If the two cysteines are in different protein chains, the disulfide bond between them forms a bridge joining them together. If the two cysteines are in the same protein chain, a loop is formed. An example of the importance of disulfide bonds is seen in the production and structure of the protein hormone insulin, which controls blood sugar levels in the body. Insulin is initially produced as a protein called preproinsulin. Enzymatic removal of 24 amino acids, highlighted in gray, and formation of disulfide bonds between cysteine amino acids produce proinsulin. Regions of the protein are identified as A, B, and C chains. Notice that the A and B chains are covalently bonded to one another by two disulfide bonds. A third disulfide bond produces a hairpin loop in the A chain of the molecule. The molecule is now ready for the final stage of insulin synthesis, in which the C chain is removed by the action of protein-degrading enzymes called proteases. The active hormone, shown on the bottom, consists of the 21 amino acid A chain bonded to the 30 amino acid B chain by 2 disulfide bonds. Without these disulfide bonds, functional insulin molecules could not exist because there would be no way to keep the two chains together in the proper shape.